neurosurgical imaging. His team, which consists of 15 trainees and engineers at the Neuroimaging and Surgical Technology Laboratory at the Mentor Neurological Institute. They developed and used computerized image processing techniques such as nonlinear image registration, model-based segmentation to automatically identify structures within the human brain. He has made significant contribution in the development of innovative software tools to align, segment, and analyze medical images. He invented original intraoperative ultrasound techniques to correct for brain shifts in imaging navigation during neurosurgical procedure. He's also pioneered groundbreaking computational methodology and mathematical modeling techniques to study the brain structure and function using neurosurgical imaging, predominantly in the neurodevelopmental and neurodegenerative disorder. He has published over 380 peer-reviewed journals. He has published over 380 peer-reviewed journal papers and over 290 conference papers and abstract. Has about 118 Google High Index scoring. So this is just a brief about him. And today he's going to be talking to us about low-cost image-guided intervention. This whole seminar will last between 45 to 50 minutes. And then after we listen to him for the first 30 minutes, and then we go to question and answer sessions. So with this background, I would like to hand over to Professor Liz Collins to continue to start the lectures. Professor Collins, are we together?
I'm here.
Oh,
Can
thank
you see me?
Yes, we can see you.
All right, and you can hear me as well, so that's good. So thank you very much for that kind introduction. That was very nice. So today I'm going to talk to you about a software package that we've developed in the lab for image-guided surgery, and in particular, image-guided neurosurgery, and even more specifically, ultrasound-guided neurosurgery. I'll give a talk for some 25 minutes or so, and then we'll show the software in action in a bit of a demo with one of my colleagues here in the lab. And I'm actually in the lab, so as soon as we finish the talk, I will spin the camera around to show you the rest of the lab and to actually do the demo. And if things go well, it should be pretty nice. So I usually start my talks, and one thing to note is that I'm a biomedical engineer. I'm not a clinician. So one of the usual first slides that I include in my talks, which is going to be very familiar to all of you, is that medical images are windows into the anatomy. We can use from very old X-ray, the very old invention of X-rays in 1895. These 2D projections are essentially the only way to view internal structures prior to surgery up until the very early 1970s. And then since then, we've had the development of computed tomography, MRI, positron emission tomography. We can combine MRI and PET together to see function on top of anatomy. You can see here, can you see my cursor?
Yes, no? We can. Try again.
We can see, you can see your slides, sir.
All right, so we can see the slides, but perhaps not the cursor. If I go, let me just check here. If I
yes, we see it now. It's a black arrow, right. Yes.
And so here we can see with the contrast medium, you can have beautiful images of the vasculature, and you can combine as well functional data from functional MRI on top of anatomical MRI to see again function on the anatomy, or you can use ultrasound to be able to image the anatomy as well. And of course, all of these imaging techniques provide windows into the anatomy that are important for surgery and allow the surgeon to see the pathology and to plan the surgery. In our lab, we spent a very long time developing different techniques for image processing. So just to give an example of that, here is a 3D vasculature that's been extracted from a CT angiogram. And we can also extract the cortical surface from the same patient, from an MRI of the same patient. And you can see how the vessels fall within the sulci of the cortex quite nicely. And of course, surgeons will use these, the sulci and the blood vessels as landmarks to help guide them in surgery. Now, the images are all very pretty, but they're only pictures of the relevant anatomy and the function. The surgeon must use his extensive knowledge to map the image information onto the patient. And this requires a coordinate system or a frame of reference. And in the past, for neurosurgery, in any case, it's been provided by a stereotactic frame. And this frame was attached to the head during the imaging, and the frame was then used as a coordinate system during surgery itself. So we want to achieve the same goals of accurate localization and guidance, but we want to do so without a frame. And so we've been developing for the past 20 years techniques for frameless navigation, where our goals are to have accurate target localization, accurate target coverage. We want to avoid sensitive or eloquent tissue. We don't want to damage any tissue unnecessarily, minimizing patient trauma. We want to track the patient, the anatomy, and the surgical tools. And we want to display all these results to a surgeon. So this is going to be a bit like a GPS for your car. So the frameless navigation allows you to plan your trip through the surgery and to guide you on your surgical journey step by step along the way. And so the frameless navigation has the same goals as the frame-based stereotactic surgery, but we're going to do so with other tools instead of using a frame. And the tools that we're going to use for neuronavigation are threefold. At the beginning, we need a camera. Here is a stereotactic infrared camera that's going to be able to track infrared reflecting spheres that are attached to tools. Here's a 3D pointer and attached to a reference device that's attached here to the Mayfield clamp that's attached to the patient. So this reference, the Mayfield clamp and the patient are considered to be a rigid body. And so we can compute the transformation between the patient through this transformation and the tools. And the last item that we need for the surgical, for neuronavigation is going to be a computer on which we're going to display all the images and do all the computations that are necessary. So the first task is to achieve this patient to image registration. And in order to do so, we have to identify anatomical points on the skin surface and identify the same points in the MRI or CT image that was acquired preoperatively. So here you can see one of the surgical staff. There's identifying points with the 3D pointer, and there's somebody else that's at the computer screen that's identifying the same points on the data on the computer. If we have more than 3 points, we can use a least squares transformation to identify the three-dimensional spatial transformation that's going to map from the images to the patient and from the patient back to the images. And this allows us to do what's shown here in the video. The surgeon is using the 3D pointer that's tracked, and it allows him to see with great precision, great accuracy, where he is with respect to the patient. So here in this little loop, he's just pushing the pointer, I believe, on the skull surface, and you can see the edge of the skull, and you can see the tissue that's just below. But this video also shows one of the classical problems that exist in the commercial systems. Is here you see that the surgeon has to change his view and change his focus from the surgical field here to the computer, and has to look away at the computer. And of course, while wearing loops, this can be difficult and takes time and change of focus and may add to fatigue. So we'll come back to this later because this is something that we've addressed in our research. So as mentioned with the title slide, we have this software that we've developed called IBIS. So two software engineers in my lab, Simon Drouin and Anka Kochanowska, have developed a full neuronavigation platform over the past 2 decades. I want to underline that this software is all open source. It's completely publicly available, and I'll put a link in the chat at the end of the talk so that if you would like, you can download the software and play with it on your systems in your labs or in your work. It's important to know that it's free software. It's open software. It's free. It's worth every penny that you pay for it, and you get all the service that you pay for. which means that we don't offer any service to help. There's some documentation, but you're going to need some people with some tech skills. You'll have to collaborate with some biomedical engineers to be able to probably get the software up and running, read your data and such. So here we show the one corner of our operating room, where here in pink is the standard FDA-approved Medtronic navigation system. And just beside it in blue is our IBIS neuronavigation system. And so you can see that we use similar cameras, similar infrared cameras. The computer system here is on this nice pedestal that's being held. Here we have a $100 cart that we bought from a hardware store here in Montreal, and we have our computer system here. The systems, both systems will see the same infrared reflective spheres on the tools and on the reference for the patient. And they will both use the same calibration or the same registration that's been computed at the beginning to align patient to image. And so with IBIS, one thing that we can do that many systems, commercial systems can't do, is to combine multiple modalities into the same image for viewing by the surgeon to plan the surgery and then as guidance during the surgery. So here we see in gray, the MRI, in color are an fMRI overlay of a particular task. This is for a patient with a right frontal glioma. Here in pink, we can see the tumor. So actually I can point here on the larger image. So we see the tumor in pink. We can see diffusion weighted tractography data showing the white matter tracts that are near the tumor that have been displaced by a mass effect of the tumor. And here in purple, we can see the large vessels that are near the tumor. So of course, this information is important when trying to decide where exactly the craniotomy will be and where exactly will be the path to get to that tumor. But I'm sure that you as clinicians know this much better than I. So I'll leave it at that. One thing to note that in the lab, our system, when you point to some piece of anatomy and you look at the images, we have sub-millimeter accuracy, which is very good and very useful for the surgeons during the actual procedure. Unfortunately, that accuracy does not continue or is not realized in the operating room. So one of the main reasons, other than technical reasons for problems of establishing the initial registration or possibly bumping the reference, if you have a very junior surgical resident might accidentally bump the reference and then make everything out of whack. The main issue or the main source of registration error is biological. And so here we can see 4 images over the course of a surgery. This is from a paper by Navabi from 2001. It's actually one of the best examples that I found. I haven't seen better examples in more recent literature, so I always use this slide. So this is the anatomy of a large tumor in the frontal lobe just before the opening. At the second image, we can see that the dura has been, that the skin flaps have been off, the skull has been opened, the dura has been opened, and here we can see a slight change in shape. This brain will swell. During the resection, tissue has been removed, and of course, just through the effect of gravity and leakage of cerebral spinal fluid, the tissues have dropped down. And here, even later in the as the resection continues, here is almost finished, you can see that there's a huge deformation from the position of the tissue here and the position of the tissue here. And when measured, this can be as big as 4 or 5 centimeters. So we have a system that is 1 millimeter accurate. Biology throws a wrench in the works and makes most commercial systems unusable. And in fact, in our institute, we have these very expensive navigation systems that are used only at the beginning to plan the craniotomy, expose the dura, plan the path. And then after that, after brain shift has occurred, the system is moved into the corner and the surgeons don't use it anymore because they can no longer trust the pointing and seeing the images on the patient. And so one of the ways that this has been addressed in previous literature is to use intraoperative MRI. And of course, MRI scanners are very expensive. If you're going to do surgery near a very high field magnet, you're going to need magnetic compatible surgical tools, which are also very expensive. You're going to need very long cables and tools for the anesthesiologist The anesthesiologists don't like that. They like to be near their patient so they can control the physiology of the patient as quickly as possible. And also the procedure significantly interrupts this, the surgical workflow. So when the surgeon decides that the images are no longer aligned with the patient anatomy and wants to have new images, then it takes 15 to 30 minutes to prep the patient, to remove all the metal clips, traction devices, move the patient into the scanner, another 10 to 15 minutes to acquire the images, and then 5 to 10 minutes to move the patient back out of the scanner onto the surgical table to restart the surgery.
Mm-hmm.
And this 45 to 60-minute delay is very long in surgery. You don't want to keep the patient under anesthesia for such a long period of time. Ultrasound is another technique that can be used during surgery. And as I understand, most of you have a radiology or surgery background, so you're going to be very familiar with ultrasound. It has many advantages. It is inexpensive, intuitive to use. There's no need to move the patient. There's no need to remove different surgical clips or traction devices. And there's very little interruption to the workflow. Essentially, the surgeon asks the scrub nurse for the ultrasound probe, which is placed in a sterile plastic bag that we can see here, does the imaging using saline to establish an acoustic coupling between the probe and the patient anatomy, and then hands the probe back to the scrub nurse. All of that takes 2 minutes, so much less than the 45-minute to 60-minute delay for MRI. But ultrasound is not perfect. There's a certain learning curve that's necessary for surgeons. It has a very limited field of view, and the images can be difficult to interpret. Although there are surgeons, as in particular neurosurgeons, that can use the ultrasounds directly to do their surgery. So I'll explain in a few minutes how we use the ultrasound to correct for brain shift in a moment. But I just want to remind you that with our system, which is this open system that has this plug-in architecture, we've been able to develop tools to acquire the ultrasound data, store the ultrasound data, compare it to the preoperative data, and more importantly, we've been able to use it to register data between the anatomy of the patient during the surgery and the preoperative data before any brain shift has occurred. And also, we've integrated augmented reality visualization into the software. So I'll show you a few pictures of this in a moment or two. So for the operating room setup, we see the IBIS workstation, which is here with the display, the tracking camera. On the display, we can see the images like we saw before. But recall that I talked about this problem of having to look away from the patient. In our system, we can actually inject the images, inject the augmented reality images into the microscope, so the surgeon can continue to look at the surgical field through the microscope and does not have to look away. So ultrasound is usually acquired in 2D, and while 3D ultrasound is possible, the size of the head, which is maybe 2.5 or 3 centimeters by 3 centimeters, is quite large and is often used for abdominal imaging, but is quite big to use through a craniotomy in the brain. And so we usually use smaller 2D probes to acquire two-dimensional images that we reconstruct in 3D like this. And this is showing a 3D reconstructed ultrasound and comparing it to the MRI. So you can see that the ultrasound shows the true anatomy at the point in time when the images were acquired, but the images have different contrast and show different information from the MR. And so the question that we asked in the lab is, can we use these ultrasounds to correct the preoperative MRI, to warp it so that it is deformed as the tissue of the patient during surgery? So one of the first postdocs that was in my lab over 20 years ago worked on this technique. And the technique that she worked on was to take an MRI, to segment the MRI, to extract gradients or edges from the MRI, all with the goal of producing a pseudo ultrasound. And with that pseudo ultrasound, we could then, whoops, let me go back. We can then take the ultrasound image, which was here in blue, and we can map it to the pseudo ultrasound there, and thus create this deformed image, deform the MRI so that it looks like the deformed tissue at the on the case. We've worked on this technique and refined this technique multiple times. So Laurence Mercier, another PhD student in the lab, worked on this technique, building better pseudo ultrasound devices, pseudo ultrasound volumes to improve the registration between the pseudo ultrasound and the real ultrasound. We've worked on vessel registration using Doppler information from the ultrasound and MR vessel or MR vascular information to do the registration. So here you can see bright vessels from the MRI, and in color, the Doppler information. When used for registration, when using these as features for registration, we used a filter to extract the center lines in the vessels, a very simplistic registration procedure to align the vessels together. And so here we can see that the MR vessels are in red, are aligned on top of the blue ultrasound vessels. And we can get from prior to doing the registration, we have almost a centimeter of misregistration to under a millimeter of misregistration. Another postdoc in my lab worked on a number of statistical mapping techniques to look at different ways. So we don't have to actually segment the MRI in advance. We can actually use the raw MRI and the raw ultrasound data. And he developed 3 different statistical image matching similarity functions that can be used to do this. As I mentioned earlier, Simon Drouin was one of the original developers of the IBIS software. He did a PhD in the lab as well and worked on augmented reality and the correction for brain shift. And these are re-showing some of the images that I showed earlier. But just to show one of the images that he did for the ultrasound, he developed, sorry, not for ultrasound, for augmented reality, we were using volume rendering to do the visualization. And so Simon built a technique called Prism. So it's a software package that sits on top of a GPU, a graphics processing unit. It's also open source, and it's also available within IBIS, and it's also available by itself, and allows you to produce different types of imaging. For example, in this one where we have this volumetric carving of the data, which allows you to query the data. What's the big contribution of Simon was that these are the only 10 lines that were required to do this type of visualization. Not only that, these are lines of code that you can actually type in while the whole IBIS program is running, and you can change the visualization if you wish, or you can have it linked in with a graphics user interface and change different parameters, such as the size of this cutting ball, if you wish. But it's just to show that the Prism library is very easy to use, and while the underlying code is complex, the use is very good. Here we have another visualization tool where we can show, as you're moving around the 3D pointer, you can actually use that to query the data here with a simulated lantern showing the different blood vessels in this vascular tree. We can also use that to identify the feeding arteries and the draining veins for arteriovenous malformation. This is important to decide which vessels to clip before removing the arteriovenous malformation. And we also have another example here of augmented reality, where we're showing a video image from a video camera that's shown here, and we're showing the augmented reality tumor being shown with the same view so that the surgeon can design the most minimally invasive or the minimal opening for craniotomy for this particular surgery. And this showing the last work, last examples of one of the students, actually not the second to last example, of one of the students working on the imaging for tumors. So here we can see the ultrasound in kind of these hot metal gold colors, the gray showing the ultrasound. You can see that, sorry, the gray showing the MRI. Here, the bright rim of the tumor does not correspond to what we see as the bright rim of the tumor in the ultrasound, demonstrating this brain shift effect. And so here on the next slide, we'll show you the registration as it's happening. We click down here in the bottom corner and the registration happens. And you can see, let me just start that again. There we go. We click and it locks on. And you can see that the registration happens quickly and is quite accurate. And I just want to note that here that the registration, this video is not shown in real time. We had to actually put it in demo mode. So to slow it down so you can actually see something happen, because the actual registration takes about 1/4 of a second. And then here, just showing an example of the surgical view. Here, where there's a large tumor that's here, here is the tumor shown with augmented reality, but before doing any brain shift correction. Here, the surgeon was asked to put the 3D pointer, which is right here, right at the edge of the tumor. And here we can see that the pointer is entering the tumor by about 5 or 6 millimeters. After we do the ultrasound-based brain shift correction, we can see that the rendered tumor now here is right where it's supposed to be, right at the edge of the pointer. I also wanted to let you know that it's not applicable only to brain surgery. Our IBIS software, we've used it in spine surgery. Here, one of my previous students has worked on spine surgery, taking the bright edges from ultrasound and mapping them onto the edges of the vertebrae so that we could do, so we could help in the navigation of pedicle screw implementation. And a more recent postdoc in the lab worked on this as well with planning software to actually plan with the size of pedicle screws and the length of pedicle screws. And we did a porcine cadaver study where we had sub-millimeter accuracy in terms of a number of anatomical landmarks, and with the registrations all computed within 10 seconds. So I just want to finish off by kind of summarizing what have we learned through this 20-odd year process. So the first is that navigation must be used to account for brain shift or tissue movement in order to maintain accuracy during navigation. The navigation systems, the standard navigation systems do not do so. Maybe the newest ones in the last year have started to incorporate it, but in the last few years, I hadn't seen very much that's actually doing the correction. Multiple registrations can be used for brain shift, but they need a bit more work to improve their accuracy, their speed, and their robustness. Augmented reality appears to be very useful in surgery, but we need more work to see exactly how it should be used and how we can improve the visualizations and how we can improve depth perception so it doesn't look like we're just putting a tumor on top of the tissue. We want to actually see the tumor in the depth. And finally, the biggest thing that I found over reviewing our last 20 years of work is that we really need to evaluate the clinical efficacy and usefulness of this brain shift correction during surgery. And we really need to demonstrate, even though I say so in every one of my grants, that patients will benefit, morbidity and mortality will drop. I actually want to try to prove that that's the case. And so that's what we want to do. So that's the end of the talk. I'm hoping that I stayed around 25 minutes. And now we're going to go to a demo with Etienne Léger, who is one of my research associates here in the lab. So I'm going to stop sharing my screen, and I'm hoping that you can see the camera on a large format. I'm going to do that. Hopefully you can still see me. Yes, okay, that part's good. Give me 1 second while I move the cart. Go. So this is Etienne.
All right.
All right.
Pleased to be with you today. So I think you went a tad over on time. So I'll try to make, so we have 2 demos actually. One is with the ultrasound registration feature of IBIS, and the other one is the augmented reality. So I'll try to make those the demos as quick as possible. So here, in terms of material, what we have here, this is a phantom of a spine that's submerged in water, right? So usually you would, what we're trying to emulate here is this would be a patient. So in this case, navigating to any specific location on the spine is very easy because you can actually just see through the water. But on a patient, obviously, it's not going to be the case, right? You can't just see through the patient. So so here, so in IBIS, I don't know if we can see well on screen, but in IBIS we have the what Louis was showing earlier. This looks just like a regular commercial system. So you have cut planes in 3 orthogonal directions of the, in this case, a CT that was acquired beforehand, and a 3D view where we have a pointer that is tracked in real time. So it might be kind of small here, but you can see the pointer moving in space relative to the spine. Oh, actually, the spine is not shown. Let me just enable this. Here, so relative to the spine. So we have the pointer and it's moving in space, and the spine model is here in space as well. But in this case here, it's non-registered, like it would be if, you know, anything is shifted, or like Louis was saying, because perhaps, you know, for any reasons, for anything that happens during surgery, you might lose registration or lose accuracy in the registration. So in this case, what I'm going to do is acquire an ultrasound suite. So I think we saw a screenshot of this module in the previous presentation. So we're just displaying on screen the live view of images as seen by the ultrasound probe. And I'm simply going to acquire a sweep of one of the vertebrae. In this case, what we're emulating in this demonstration is if we want to register one vertebral level in particular. In this case, it's L2. So I just do the acquisition here. Like you saw, it takes, I don't know, maybe 20-30 seconds. Then I'll click on run registration. There's a bunch of parameters that you can tweak to make it work in different types of situations, and make sure it works for your specific data set. And then you, when you click run, it will run for, as you just saw, maybe, I don't know, 45 seconds.
We get close just to show the fit.
Yes, I'll move on this side perhaps. And then once that's done, we need to move this. Need to unzoom a little bit. So here we have the spine, and now that it's registered, I can place the pointer somewhere on the phantom, and I'll see this location displayed on screen. So I'll see the pointer point on the images at the same place where I'm pointing on the actual real patient. So this can be used to navigate to any specific location, or to plan for how this can be used as well, is to plan an insertion path, right, within the vertebra. So this is the first demo, and you can see how overlaid on the same model you can see. So it may be hard to see now because it's a bit small with the camera, but in white we have the CT, and in red we have the ultrasound reconstructed volume, and we can see that they match very well in this case. So it's the registration is accurate, and this is a very easy to see. It's very easy for the surgeon to see that the registration actually worked during the procedure, and if it didn't, if you can see that it's off in the images, then you just require another sweep, right? So maybe you'll add another 30 seconds or a minute to the procedure, but it's very easy to detect when something has gone wrong and to correct for it. So this is the first demo. The second demo, again, I'll try to go quickly. So we have a tablet computer. And the same IBIS software running on the desktop machine. So in this architecture, we actually have, whoops, let me make this full screen. Let's just have-
Stretch it down.
Computers. Okay, and here I'll put in track mode. So now what I have here is so this is a CTA that was acquired. So this is now in this case I'll move this aside so that you can see. This is a 3D printed head of an actual patient that was operated here at the Institute. And so this patient had a CTA done before a surgery. And what we can see on screen now here is the live video feed. So you can see if I move my hand underneath, I can see my hand in real time. And overlaid on top is the segmented vessels. And what this enables you to do is, well, first you can see structures that you wouldn't see right under the skin surface. And also it makes it very easy to look at anatomy from different perspectives. So if I move around the patient, I can perhaps better see which, if for instance, this could be used by a surgeon if you're hesitant between two different approaches to reach a specific target, you could try to look at the anatomy from different perspectives and try to make sure that you're not intersecting with some other structure that you want to not disturb on the way, for instance. Yeah, so this is the second demo. And what this enables you to do as well is, the interface is all customizable. So in this case, we're seeing vessels, but you could, from the interface of the application itself, turn the if it was, in this case, it's an arteriovenous malformation, so there's not much more than the vessels that we might want to look at, perhaps. But if it was a tumor case, you might have the tumor and the vessels, and perhaps, as we saw in the presentation before, maybe fibers extracted from DTI images. So there's all these different sources of information that can or cannot be displayed, and you can change the rendering on the fly, change what is displayed and where exactly, and you can focus on some area. So this hopefully gives a way surgeon to look at the anatomy in a very intuitive way during the procedure. And the iPad like this can be draped in a sterile bag, as we saw in the presentation before was done with the ultrasound probe. So it's also very easy to sterilize and to use intraoperatively. And so we hope that with both the ultrasound and the correction intraoperatively and this perhaps more intuitive visualization, we have a system that both is low cost because it's the software is free. Like you said, we don't provide much support, but there is documentation and it is available online. So the software itself is free, and all the tools and everything we build, we also try to make available as much as we can. We also sometimes use parts from other manufacturers that we can't necessarily that can't be replicated. I mean, potential users will have to buy them off the shelf as well. But
So,
yeah,
all right.
that's the demo.
So one of the things that we wanted to put out, that we are currently putting together a low cost system, or a lower cost system, but a low cost system. So the system that we have here has going around here has a BK 3500, which is about $300,000. Excuse me, $300,000 Canadian. We're using the Atraxis tracking system, which is about $50,000 Canadian, and then a pretty powerful computer, excuse me, with a GPU. So all around, it's about $350,000 to $400,000, which is not an inexpensive system. The work that we're doing now is to use these new ultrasound probes that have a small little controller box like this, have a USB connector like that, and an ultrasound probe like this, and these cost $3,000. So we can drop the cost of the ultrasound from 2.5 or 300,000 by a factor of 100 down to 3,000. We have also a camera that's similar to the Atraxis, but it's much smaller, sold by the same company, and it's about $5,000. And then all of the software is now working very well on a laptop computer. So we have a system where the hardware is approximately $10,000 worth of hardware, give or take some. And so we're trying to get everything. Well, we have everything working with these low-cost items, but we'd like to be able to have people evaluate it and see how these can be used. And so we are looking for collaborators to do this. And of course, so given the software is all open, you are also free to do whatever you want on your own without collaborating with us. But of course, we would like to collaborate. So that's pretty much it for our demo. So I'm going to come back to where I was so I can sit down. And that's the end of the presentation and demo.
Okay, thank you very much, Professor Collins and his team. This is really quite wonderful. But well, I would say the lecture was a bit technical, but with the demo, it simplified a lot of things and has become more clear. With this, we have come to the end of the first segment, which is the presentation and the demo, and then for the next 10 to 15 minutes, we'll be looking at the question and answer session. But before we go to that, I would like to give a brief summary of what Professor Collins has presented. He summarily talked about IBIS, that is intraoperative brain imaging, and he was highlighting the importance of the system, in the sense that if we are going to be using intraoperative magnetic resonance imaging, it's too cumbersome, it takes more time. He highlighted and it's more technical for the anesthesia team. And one more important thing he did not add, probably because he, as he's mentioned, he's an engineer, the risk of infection is very high because all the gadgets, they may push the incidence for post-operative infection to become much higher. So this system that is much more friendly, risk of infection will actually drop. Then you talk about virtual reality, augmented reality, and then how they are being used in the demonstration aspects. And one thing that catch my attention very importantly, and that is very applicable to us, is the use of augmented reality in pedicle screw insertion in spine surgeries. Really, it's a very it's a disaster when you pass.
I'm sorry?
Network connection, Louise.
So it's not only at the MNI that we have difficulty with our network.
No. Okay. Dr. McGill, are you still there?
Okay. Hello. I missed, but I'm back. Doctor.
All right, great.
Yeah.
Yes, I'm back. Yeah, I'm talking about the augmented reality in pedicle screw insertion. It is very important and very much relevant to us in Africa. And personally, I'll be looking forward how we can get that technology down here. And then I think with this, we can go to the question and answer session. I could see one question from, okay. There's a question from Dr. Mariam Aliyu, that is asking about what is the cost of the navigation system if we compare with the IBIS. I don't know, Prof. Collins, if you can answer this question.
Yes, so the navigation systems, the commercial navigation systems are quite expensive. They run from, actually the navigation system without ultrasound runs at the cost of, I don't know, from $500,000 to $750,000 US. And gives you the navigation and the tracking. When you include the ultrasound with that, that'll add another 300,000 for the device and probably another 100,000 for the license. One of the big sources of cost, I think, for the FDA approved devices is in fact the cost of acquiring FDA approval. So the approval process is extremely expensive. And so while we can develop a system that will work and that we use here under in a research environment under REB approved conditions. The system that we have, as I said, the current system is $350,000 worth of equipment and software, but I think we're going to be able to achieve very similar results with a much lower cost ultrasound. So for less than $10,000 of equipment, we'll be able to use the similar system, but only in a research environment. It won't be able to be used as the clinical standard until we get some type of approval for that. And that approval takes a long time and is very expensive in North America and Europe. I have no idea what the costs are like in Africa.
Okay. All right. Thank you very much, Mr. Collins. And Dr. Mariam, the person that asked the question, I hope he has answered your concern. If you have any other issue regarding it, you can unmute yourself and ask question directly. Dr. Mariam, are you hearing us? Okay, I think we can go to the next question. There's another question from Mr. Nuruddin Kadri, who was asking that how is the vessel segmentation done during the intraoperative period with the navigation system?
Okay, so all of the vessels are segmented preoperatively for the either the preoperative CT or the preoperative MR angio. The processing system is based on a Frangi vesselness filter. It's a second order Hessian filter that is passed through the data and highlights tube-like structures at different diameters. So you essentially would run a vesselness filter to extract all the vessels of 0.5 millimeter in diameter, then 1 millimeter, and then 1.5 millimeters, and then 2 millimeters. And then there's other software that's used to combine all of this information together into a single vascular model for the actual patient. Of course, there's other software that's involved in removing the false positive structures. There can be other imaging artifacts or other brain structures that are slightly tubular in shape that can be extracted by those filters, but are not blood vessels. And so that would be the first bit of software that's used for building the basic vessel or vascular tree. Once the vascular tree is done, you can process the vascular tree even further by trying to decide what are arteries and what are veins. And that can be done by running a geodesic distance from, let's say, from the carotids or from the jugular veins on both sides. And any part of the vascular tree that's going to be closer to the jugular is going to be judged to be a vein. Anything closer to the carotid is going to be evaluated as an artery. But of course, that has to be verified once the processing is done.
Okay, thank you very much, Mr. Collins. And Mr. Nuruddin, I hope you have answered your concern. There's another question from him asking about how will I be able to replicate this with the software and ultrasound in a hospital setting? And I will be very much interested to be involved in this. I also have the same question like him, that in terms of the augmented reality during spine surgery, if I have an ultrasound set and in my hospital setting, like in Nigeria, how can we incorporate this procedure in ensuring that our pedicle screws are properly inserted?
So well, it's a bit of a complex problem, but it's not complex just because there's many details, but it's not anything that's impossible. So as we said, give me just a moment as I plug in my computer. I just got a low battery warning. I thought I had a fully charged battery, but there we go. These, good, we're safe. So the ultrasound probe that we're using, like this particular low-cost ultrasound probe, is I forgot the actual brand it is. It's a Telemed system. I believe it's available worldwide. The tracking system from Atraxis is a Swiss company.
Traxis, yes, but OptiTrack is an American company.
And OptiTrack is an American company. They are pretty sure that they both sell worldwide. And if we wanted to be, if you really wanted to go low cost, and if you had good friends in an engineering department, you could try to build something that looks like this. This is it's a bit of a mess with all the cables on top, but it uses standard webcam video cameras. I'm going to try to show one of them here. So this is a little video camera. So I had a bunch of engineering students here at McGill for a capstone project put together for $1,500 full tracking system, which is kind of nice. But $1,500 might be a bit much for something that might possibly break tomorrow morning, in comparison to a $4,000 or $5,000 Atraxis type small tracking camera. But if you're really interested in putting it together, or just send me an email. And if you're interested in the collaboration, please send me an email. I'd really like to establish some links for people to actually try to use the software, try to install it, try to get it to work in their institute, try to evaluate it in their labs, and then potentially, after REB approval, try to use it with patients to evaluate to see how well it can work. I think it would be very useful.
Oh, thank you very much, Mr. Collins, and surely you will see my email. Would like to collaborate.
That would be
all
wonderful.
right. I don't know, Nuruddin Kadri, can you unmute yourself if you have any other concern? Are we together? Okay, as I'm doing this, the evaluation form has been shared. Please, let's try to fill the evaluation form so that we help our organizers to see where they need to do more. Okay, so these are the questions that were asked, and I think we are coming to an end of the session. But I would like to ask Dr. Oduna if there's anything you want to say or you want to pass information to us before closing.
This has been very fantastic. Thank you both. Thank you, Luis, and also Dr. Nagashi for hosting. We had a little bit of Zoom interruptions, but we are recording this, so there may be some follow-up questions for people that were not able to join. And Luis, I'll pass it on to you. But this has been fantastic, and I'm so grateful that we both were able to do this for us. Thank you.
This was great. Thank you very much for the invitation. And
Okay.
I'm pretty sure that Etienne says thank you as well.
Yes, thank you, Etienne.
Great.
Okay, so I think we can close this up. The next seminar